Hi and hello to the first international episode of the Dive In podcast. I'm here currently at Stockholm, Sweden at Karolinska Institutu University, and my first international guest is actually Kate Stanely, coming from 3 different countries, or 2, I don't remember, but Kate is going to tell you in more details. Anyhow, Kate, I want you to first tell me, as I always start with my guests, how did you go into the field of neuroscience, embryology, and how this academic journey actually started?
Well, I didn't have any big master plan when I was younger. I was sort of vaguely interested in biology. I took the International Baccalaureate, which forces you to do 3 higher subjects. I did those in biology and chemistry. But my view was open. I applied to medical schools in Europe, and then I also applied to liberal arts schools in the States.
Before going into that, can you just briefly explain to the spectators, like, where do you come from?
Oh, yeah.
Your background?
I'm Irish-American, so my mother's Irish and my father is American, but they're expats in Belgium, in Brussels in particular, the capital, and they've been there for about 30 years, and they had their kids there, so me and my brother grew up in Brussels, and we went to an international school, so it's a bit of a mix, but yeah.
Okay, and then you went into IB school?
And then I did the International Baccalaureate in high school in Brussels. So that's a 2-year high school course where you, as I said, take 3 highers and 3 standard courses, and then you use those to apply to universities.
Mm-hmm. And then you went for your bachelor's where?
My bachelor's was at Columbia University in the city of New York. Mm-hmm. And there I majored. So in the States you major, you don't go straight into a subject like you do in Europe. You have a focus, but it remains quite broad and quite open. So Columbia in particular has quite a sort of holistic approach to teaching, where they require that you take what they call a core curriculum, which includes philosophy courses, literature courses, music.
For how long do you need to take them? 2 years?
The core curriculum takes around 2 years, of your 4 years, but a lot of people sort of dawdle, and sometimes you'll be slapped with an art course in your final year of university. But yeah, then of course you also do a focus, and mine was on neuroscience.
Did you get a scholarship for Columbia, or it was something that you financed? Were you considered as a home student or as an international one in your case? So I have an American passport, so I was considered I don't think they call it a home student. I wasn't a foreigner. And Columbia has what they call a need-blind system. So you apply to Columbia without them knowing anything about your family's finances. And then once you're admitted, they look at your family's finances and they give you what they think is appropriate. Anybody who has a joint family income of less than $100,000 a year gets a full ride. And then there's a full ride is 100%.
100% coverage of your tuition, yeah.
Did you get any?
Yeah, I got a huge amount of aid because my family is not extremely wealthy, and I mean, we obviously are doing fine. My mother's a teacher, my dad is an independent, but Columbia is an extremely expensive school, and schools in the States are really, really expensive. So we got a large portion of the tuition.
What did they ask for your admission? What did they ask for your admission? Like what kind of documents?
What did they ask? Yeah, gosh, it's a long time ago now, but they asked for an essay, and then they had a couple different questions that they asked that you needed to answer. They asked for a CV, obviously, and the SAT. I think I sat the SAT like 6 times. It was a really difficult test for me, and you have to score quite
because
highly.
It's maths, probably. Was it maths?
It was more the timing. The SAT you have to answer each question in, like 30 seconds or something. I didn't do very well on the SAT after all of those attempts, so I switched to the ACT, which is an alternative standardized test that you can do instead, and I did much better on that, so I applied with that.
Okay, and then when you got into it, how was your feeling, of the Columbia community and general experience of studying there?
I loved Columbia. I couldn't rate it more highly, honestly. I loved that they had the core curriculum, although it was really debated at the time on the campus. They force you to read a bunch of like old white guys, basically, which they consider sort of the masters of Western civilization. And I think that was hotly debated whether or not that was sort of the right curriculum to be forcing upon students.
How were you graded, your exams, like the way Columbia is functioning?
You take about 4 or 5 courses a semester, and then as they start out as general courses where there's big lecture halls, might be 200, 300 people in a lecture hall, and then towards the end you start to take seminars, and those are usually very small meetings of about 3 to 4 hours a week where you're with 7 other students and a professor. And some of those were the most enriching because you had really close relationships with professors who are just really at the top of their game.
Why did you decide to go for your master's in Europe after Columbia?
I took a break. Well, for one, I worked so hard to get into Columbia that my first 2 years of my bachelor's degree were kind of a vacation. I didn't do very well. And then I realized that I wasn't doing very well, and I really picked up the pace, and I got my GPA to a place where I could at least get a job after I graduated.
What was the GPA in the end?
My final GPA was a 3.51.
Oh, amazing. Out of 4 to
out
0.
Of 4, yeah. And that was actually the limit to get into graduate medical schools in Europe, because I thought maybe I would want to go back in that direction. But it turned out that that's not what I wanted to do. Because I had been living abroad for so long, I couldn't get European tuition at the graduate schools, actually. I would have had to reestablish taxpayer and residency in Europe in order to get EU tuition at medical school. So I basically bagged that and started working for 2 years.
Where did you work?
Where did I work? I worked at Columbia Medical Center.
What did you do there?
I did fetal exome sequencing.
Can you explain a little bit further?
Yeah, so fetal exome sequencing is basically when a woman comes in with a pregnancy complication, or she gets her sort of screens. They do ultrasound screening in every pregnant woman. And when something is seen that's a malformation is seen in the fetus, they typically go in with a big needle and they biopsy some of the placenta. It's quite safe.
It sounds scary.
It is really scary, yeah. I remember watching Dr. Ronald Wapner do a couple in the clinic, and the women were really stressed. But, you know, he's excellent at what he does, and typically the doctors are excellent at that procedure, and the risk of miscarriage is really low now, like under 1%.
Do you have any new modern techniques that you practice now instead of that one? So
there's non-invasive methods now, which is what my PhD is on.
Oh, then we're going to come to that later on. Okay, I jumped to the conclusion. Okay, but then after 2 years of working in Columbia Medical Research Center, you decided to go back to Europe to go pursue your master's degree.
That's right.
Where?
At Oxford. I went to Oxford because it's Oxford, basically. But there was also an embryology course that they were giving there, and I was really interested in doing that. I was working as a bioinformatician, so I was working mostly with large public databases, but maybe I should back up. Population genetics uses sort of genetic information that's being taken across loads of different people and looking at frequencies of mutations. And if you see that a mutation is really lowly prevalent in a population, it more likely that it's causative of disease. But we never really got into the specifics of why a gene might be important for development, or where and in what pathway and what its role was in development. So I wanted to know that. I wanted to figure out what genes were important players in fetal development. And so I went and I did an embryology degree at Oxford.
You diverted from neuroscience to embryology. Did your love for embryology started at Columbia Medical Research Center, maybe? Your love for embryology, did it start where you were working at Columbia?
No, my interest in developmental biology really came when I had an abortion myself. So I was in a neuroscience degree. I didn't particularly like neuroscience. It was a lot of statistics and physics, or not really statistics, but a lot of physics. And I didn't take to it really at all. And then towards the end of my 4-year degree, I got pregnant, and it was an unwanted pregnancy, and so I had an abortion. And I needed to know more about it. It was a way to soothe me, I think. I got really invested in the biology of fetal development, and that's where my interest really got started, and then it just really snowballed from there. Like I used it at first to get over what I considered a traumatic event in my life, and then it really just became like an academic interest. And so I pursued it after my undergraduate degree, and I'm so happy that I did, because it's like an incredibly interesting and fast-moving field, and I really feel like I'm helping a broader problem in the world, which is, you know, how do we take care of women, and how do we have good prenatal care?
Okay, we're gonna come to what your PhD is about. Tell me more about your experience at Oxford. How was that compared to Columbia, teachings and exams? And
yeah, I think
was it harder? You know, there is always a rivalry between American and British university in the terms of rank and the teaching quality. So what would you say about that?
Do I have to pick one? Do I have to pick Oxford or Columbia?
No. Just
they
compare.
were very different. They were very different courses. My bachelor's degree, as I said, was a mix of humanities and science. Oxford was very specific. I was doing a master's in embryology. We had weekly essays that we had to write. It was much more intensive. And fast-paced, and also crammed into 12 months.
A year.
Yeah.
Of master
well, yeah. So a lot of the masters at Oxford are 9 months, but we had a 12-month extended master's degree, which allowed for a 3-month research period. I think Oxford, they definitely encouraged you to write for the examiner, which I found quite peculiar. I thought that their teaching method was actually slightly archaic, like a little bit traditional, yes. A little bit backward. It didn't really push for super critical thinking, I found.
It was more like readings and doing research theory other than practical matters?
There was a lot of reading, a lot of research, and a lot of terrific lecturers. I mean, I don't think that I've had such great lectures in my life than from the people that were on the course at Oxford. But in terms of the way that we were graded, it was very to the rubric, and it was very strict. And I found that
Nina just told me that you only have one time to pass the exam. Otherwise, like the second time, they're gone. That's it. You're done.
Yeah, totally. And you have to like wear funny clothes and stuff. So
did you go into rowing? Because everybody goes into rowing. And I have to ask that question because every person I've interviewed from Oxford is like, yeah, I did the rowing classes and then the balls and then.
No, I didn't row. I wasn't a rower.
Okay. Did you have any social activities other than just academia?
Yeah. I mean, I met a group of incredible people. They're all literature PhDs.
Okay.
I know Nina was friends with her embryology folks and more sciencey friends, but I've never had a huge amount of sciencey friends. So yeah, I met a bunch of people. It was also COVID. I mean,
the period was. Were you in England the whole time?
Yeah, I was.
And how did you take your exams? Was it online?
It was online, yeah.
That messed your experience a bit.
It did a bit, yeah. I mean, I do a lot of computational work, so it's usually on the computer. I think people who were in the wet lab felt really sort of like, you know, they were disappointed, but it was fine.
Okay. Then we go further, and then you didn't stop with just your master's degree. Then you wanted to do a little more, actually a lot more research. Yeah. And then you came to Sweden, to Karolinska, the place we're at now. So why did you choose Karolinska in Sweden?
Well, Karolinska is a big name. I mean, I know that it's a world-renowned university, and so is KU Leuven, which is where
in Belgium,
yeah, which is where my main house is. But I was really looking at the science. I deep dived into the PIs that had posted the PhD, and I liked what they had been doing, and I liked the work that the lab was doing.
Okay, and when did you apply? What was that year, 2000 and?
Yeah, so I was at Oxford for a year, 2019 to 20, and then I applied in, August and started in September.
And did you apply to multiple universities, or it was just specifically only this one?
I also had a...
Did you test your chances otherwise?
I applied for a PhD at Oxford as well.
And
got that, but I decided to do this.
Yeah, like Nina. It was like because... We talked, but I think it would be also good for you to say that in English, that this is not just a regular PhD, it's a job.
Yeah, it is a job.
It's a job.
Yeah, it really is like a 9 to 5. Some days it really feels like that. Yeah.
So what are your main responsibilities? Can you tell me more about your PhD thesis and what do you do now?
Well, I wake up and I have coffee. That's like a critical point of every morning. And then I work from home quite a bit now, especially when I was in Leuven, because I live in Brussels and there was a commute. But when I go in, it's a lot of chatting, catching up with my students. It's funny because with my students, they're not my students, but with like fellow students. I always find it difficult because, yeah, sometimes I call them colleagues, sometimes I call them PhD students. It's a difficult, it's not difficult, it's just it's a mix, you know. Sometimes you feel like you're working with them, sometimes it's an independent, more of a research environment, but yeah, a lot of checking emails, and then I really get into it. I mean, I'm always on the computer, I don't have any lab work, so...
Okay, and your PhD thesis, what it is about?
It's non-invasive prenatal testing. So I basically work with maternal blood samples. So what we do is in Belgium, 80% of women who are pregnant will receive what's called a NIPT test, non-invasive prenatal testing. And they take a blood draw and then they isolate what they call cell-free DNA. And cell-free DNA is just DNA that's been released by dying cells. So you have inside each cell you have a nucleus, and inside each nucleus you have your entire genetic code. And when the cell dies, enzymes within the cell chop up the DNA and they release it into your bodily fluids. So you have sort of what like trash DNA, you could think about it as, in all of your bodily fluids. So urine,
and you take those.
Yeah, and you take it from the blood. And when you're pregnant, the placenta also has cell turnover, and so fetal DNA will find itself in the maternal bloodstream. And you can use that fetal DNA from the maternal bloodstream to diagnose a host of different disorders.
Mutations or some kind of genetic disorders?
You can look for like large sort of like an increase in the amount of fetal reads, which usually that map to a specific chromosome. So that shows you trisomy 21, which is Down syndrome. Yeah,
which was the most common, right?
Yeah, so common trisomies, but you can also see partial deletions, which are associated with all different disorders. And what I'm doing is I'm trying to broaden the diagnostic scope of
diseases.
Of diseases that you can detect.
Amazing.
Yeah.
And at what year of your PhD are you now?
I'm in my third year.
Okay, and to what conclusions did you came? Any breakthroughs?
Yeah, so we're getting close to being able to identify a whole host of different pregnancy complications, especially
when... What about autism? Is it possible to do that with prenatal diagnosis?
Yeah, but autism is typically considered a monogenic disorder. So there are, not all of the cases of autism are monogenic, but some of them, some proportion of autistic children have mutations and genes that are associated with the disease. And you can diagnose those non-invasively if you'd like. There's non-invasive prenatal diagnosis, NIPD, where you call variants, so call mutations that are associated with the disease. But typically you might identify those later post-birth because a child develops symptoms, because autism doesn't develop until a little later on in life. So if a child develops symptoms, you might get given an exome panel, like a panel where they sequence your child, and you might get a genetic diagnosis. But prenatally, autism isn't typically diagnosed.
And which are? What diseases?
Really severe. So you can go for carrier screening. So if you're too there's a bunch of different services you can have, right? So, and it depends on the country and the regulations, but you can have what's called carrier screening. So before you are pregnant, you and your partner go in for a test where they sequence your genome and they look for pairs of mutations between the two of you that, if inherited together, would cause disease. And then you can, if you are both carriers for mutations that, if inherited, would cause severe disease, you can then go down the route of IVF and choose embryos that don't inherit both of those variants.
And did you ever do, because I've heard about that, for example, if couples have unmatched blood groups, that can also impact the pregnancy, and you have to maintain it. Yes. Have you ever dealt with that? Like, for example, if I have A negative and my partner has A positive, the fetus might be,
Really?
Yes.
I've never heard about that.
Oh, you see?
No.
Yes, I've heard that. Yeah. Okay. Anyhow, back to your subject. So, what is happening now with prenatal diagnosis, and what we can expect in the future?
Usually you only get a genetic test or a prenatal test other than this NIPS test, right? Which is like searching for like very severe disorders. Usually you only get a genetic test if there's an ultrasound anomaly, right? Or you're a known carrier beforehand. But there's a sort of a very contentious new line of prenatal testing that's being developed predominantly in the States, really. It's it only has happened in private clinics that are offering polygenic risk scoring for non-medical traits. And there's been a lot of ethical concerns around that because obviously polygenic risk scores, when, unlike autism, what we were talking about, these monogenic diseases that are quite severe and caused by highly penetrant variants in a single gene, you have disorders that are called complex disorders that are caused by lowly penetrant mutations across the entire genome that in aggregate cause disease. And now there are tests that assess your risk for those. For a whole,
And what's the percentage of, them showing you correctly the situation? Can they be wrong?
So it's a risk. So it's also not.
For example, you tell me, Masha, you have like predominantly this, genetic mistake. Maybe it's going to happen with the baby. Should I believe the test and decide not to go for the pregnancy?
Yeah, it depends, right? It really depends what it is, how high the risk is, and how severe the disease is, right? Okay, but that's disease. Now we're talking about non-medical traits. So you have the opportunity as a parent to go into a clinic, or you did at least. I think it actually stopped, so you'd want to double check me in the regulations in the States right now. But at least for a time, there was private clinics in the States offering polygenic risk scores for non-medical traits. That means educational attainment. That means height. That means, these are traits that really shouldn't be selected for in a clinical setting because they're not medical. But it was being offered. And the added layer of...
Is it done with that? Or
I believe that those services have now been suspended.
I hope so.
Yeah.
It's not ethical. It's
really bad. It's a very difficult line, yeah. And on top of that, those polygenic risk scores are trained on data that's predominantly generated within the Caucasian population. So those scores and those risk assessments really only work in the Caucasian population. So even if you had a family of color who wanted to go and get this service, it wouldn't work for them at this stage.
Okay.
Yeah.
Back to, I heard, actually, you sent me the text where your mentor professor talked about stillbirth. Do you deal with stillbirth a lot at your PhD? And can you talk about what is that, to explain how does it happen and what causes it?
There's a rubric really that's out there. There are a couple of different international systems that try to categorize the causes of stillbirth. You might think of acute trauma that could cause a stillbirth, maybe maternal infection that might cause a stillbirth. But what's really surprising is that actually a lot of stillbirth is unexplained. And so back when I was working at Columbia Medical Center after my bachelor's, I worked with David Goldstein and Ronald Wapner to sequence a large cohort of stillbirths and look to see whether or not there were genes associated, underlying genetic causes of stillbirth. And we found that there were definitely specific genes with mutations in them that caused disease, known genes that were associated with disease. And could that be tracked
before if they did a prenatal testing or something like that?
Well, a bunch of them were also not associated with disease, and that's probably because we haven't done a lot of sequencing in the stillbirth population. But again, right now we don't offer exome sequencing prenatally, so you're never gonna we're not screening for that currently. In my PhD currently, though, we're looking at ways in which we might be able to detect it non-invasively.
Okay, and how are you satisfied with the experiments right now and everything you do right now?
It's good. It's moving along. It's always there's waves in a PhD, and you have to kind of ride them. But yeah, I'm kind of, the wave is building at the minute, so it's good.
Okay, can you tell me some interesting things about the prenatal testing or your PhD? Something that can be really interesting to our spectators. Something that you found out, or maybe something we should know, like that should be publicly, available to us to know.
I mean, it's good to just know what's out there, as a woman and a man in the world, maybe having a child one day, it's just good to know which tests are available for you and why you might want to have them.
You want to talk about them?
Well, I have mentioned most of them.
Okay. Anything else to mention?
No, I don't think so.
Okay, Kate, what are you planning later on? Like, I mean, when you finish your PhD, when will you finish? In a year?
I'll finish in a year and a half, yeah.
Yes, and after that, are you going to go to be a lecturer, stay in academia, work in the industry? Where do you see yourself after this career in embryology? I don't have a huge plan yet. I'm just trying to get through my PhD and hopefully publish as many papers as I'm supposed to publish. How many papers? Because I asked Nina, she said 3.
Yeah.
But probably she's expected to publish more, but for you?
Her host is Estonia, so she has to publish 3, but I have to publish 1 for KU Leuven and 2 for Karolinska, so 2 in general.
And do you commute between Belgium and Sweden all the time?
Well, trying not to take too many planes now, but I do have to commute back and forth.
How do you like Sweden? And what do you like the most here?
It's so cold here.
Everybody's like, so winter, don't come winter.
No, but I love it, honestly. Yeah.
What is the prenatal testing in Sweden? Like, how do they do it?
What is the prenatal testing in Sweden? Well, they offer NIPT, but only about 30% of the women use it.
Is it, I didn't get it, is it invasive or non-invasive?
It's non-invasive, so it's the blood test that I was talking about, yeah. And they look for like a gross over or underrepresentation of reads.
And women should do that one at what period, like what week of pregnancy?
It's between 12 and 16 weeks of pregnancy.
Okay.
Yeah.
Okay. Anything else?
Well, we're hoping that, like, my PhD is really focused on making, like, can we use these cell-free DNA fragments to identify growth restriction or preterm labor or preeclampsia or...
Preeclampsia? Can you explain them?
Yeah, preeclampsia is hypertension in the mother caused by sort of a failure to remodel uterine arteries. And so you get a lot of high blood pressure and it can be really detrimental to the pregnancy. Yeah, so there are all of these complicated things that can happen during pregnancy. We don't talk about them very much.
No, nobody talks about it at all.
No, women are kind of like, encouraged not to even tell people that they're pregnant until the second trimester.
Yes, actually until they're like 3 months pregnant.
Yeah.
Until they're like, don't tell anybody. Is that supposed to be like that?
I would encourage women to say it. I mean, I think it normalizes the fact that pregnancy is really complicated and stuff can go wrong.
Because everybody's telling us, like, maybe you're gonna, maybe something bad will happen, and you made us all happy, you're so happy, and then something bad happens, and then what we're gonna do next, It's a huge grief and sadness.
Yeah, so and it's a secret, and why does it have to be a secret? There's no shame.
We should encourage women, tell immediately when you're pregnant and be positive that everything's gonna be fine, because they actually tortured us in the brain that it will go badly. So
and if it does
those
go badly,
3 months you're under stress, thinking that it will go the wrong way. Actually, it's the whole point of the message, like, think that it's going to go the wrong way, so be silent.
Right, I know. And I think a lot of that is because we don't have good tests right now. We don't have ways to monitor whether or not something will go wrong, and hopefully that changes in the next couple of years.
Kate, okay. Do you have anything else from your biography you would like to share with us?
I mean, there's a question on there that's like, how would you suggest or encourage people who are going into human genetics? I would say for that one in particular, like, there is so much data out there already. There's so many genomic databases out there, and so if you're interested in pursuing a career in genetics, go and play around with the data that's already out there and teach yourself some things and get used to ask yourself some questions and try to answer them with the publicly available data that's already out there, because there's a lot of it.
Would you encourage students to go into embryology as a field?
Yeah, it's great.
Do you think there is a lot of places for, you know, as PhD students, you're bound to discover something new usually. You have to discover something new in order to become, actually, to finish your PhD. Is it still an unsearched field for us humans?
No, I mean, people are definitely, it's a big community. There's just a lot more to know.
Okay. Kate, I always have the question for my guests, which is the message you would spread to the young people, like about education or in general, like your conclusions along the way. So what would you tell them?
Don't put too much pressure on yourself, I think, and find people who believe in you, and hold on to them, and you know, opportunities come to you as well. I think that I didn't have a fixed idea in my head. I let opportunities arise, and I followed them. I didn't have any big fixed plan, so I think that's a good, and it's worked out for me. So
not to have a plan, actually. I mean have one and work towards it. Okay, but what about dreams? And you know, your how would I say that? Like, for example, you come mostly this is going to be broadcasted to Balkan people. Like when you come from a part that's like EU country, Belgium, probably, and you speak English perfectly, there are a lot of more opportunities. What would you suggest to those kids that are not from that area, that have actually access to those institutions or databases or whatever? Yeah. What should they do?
Well, you can only get in if you apply, right? So apply. And reach out to people via email. I mean, that's cold email people. I've definitely done that a lot in my career.
People in my area think that if they email a professor, they're going to be, oh my God, disaster is going to come for their application. They're going to be rejected, or it will have an influence on the admission outcome.
Definitely, and especially email people directly. And if they don't get back to you, they don't get back to you. But sometimes they do. And if they can't answer your question, or they don't have a position open, they might redirect you towards someone else. That's how I got my first job. It's just a matter of putting yourself out there, really.
Kate, thank you so much for actually taking the time and sitting here with me. I know you have a lot of things to do with your PhD.
That's okay. Thanks for having me.
Thank you so much for being my guest. I wish you all the best with your PhD, and I hope you find the new testings and discover way more new, like a broader picture of diseases that can actually impact the pregnancy.
Me too.
Thank you so much.
Bye.